All right, welcome back everyone for another Journal Club Thursday. This is another article on tape, so to speak. This week we'll be doing a Throwback Thursday and reading the arguably most underrated publication of all time, Transgenic Force Sensors and Software to Measure Force Transmission Across the Mammalian Nuclear Envelope in Vivo. Introduction. The cell nucleus experiences physical forces in a manner that has the potential to alter chromatin conformation, gene expression, and downstream consequences such as cell identity and movement. Transduction of force is an alternative and more rapid means of transmitting signals to the nucleus than the diffusion of biochemical signals. By advancing mechanisms of mechanical signal transduction, we can potentially generate and test a broad range of biological hypotheses, including those concerning development and disease. For example, forces that shape embryonic tissues might co-regulate cell differentiation and or the expression of patterning genes, and feedback from pathologically stiff tissue environments, might alter gene expression to promote the progression of chronic inflammation and neoplasia. Many of the key methods required for this area of research, such as quantitative analysis of RNA and protein expression at single cell resolution, determination of chromatin conformation, and measurement of nuclear and tissue viscoelastic properties, have advanced considerably. In contrast, tools to measure forces experienced by nuclei are only just emerging. Recently, genetically encoded sensors to measure force transduction across the nuclear periphery have been described for in vitro studies. Model organisms expressing force sensors would be useful to define the extent to which actomyosin or external stresses that deform the nucleus are transmitted across the nuclear envelope in developmental and other physiological contexts. Recent attempts to utilize transgenic tension sensors in vivo have generated both promising and discouraging outcomes. Challenges inherent to employing force-to-resonance energy transfer FRET-based sensors in vivo need to be addressed before they can be applied robustly in animals. Mechanical connectivity between the nucleus and the cytoskeleton with extracellular matrix or neighboring cells is increasingly understood. Underlying that connectivity are physical attachments of the cytoskeleton to the nuclear envelope through the linker of nucleoskeleton and cytoskeleton LINC complexes that links F-actin microtubules and intermediate filaments to the nuclear lamina. Mammalian aspirin proteins that reside across the outer nuclear membrane bind cytoskeletal filaments such as F-actin and are anchored to the inner nuclear membrane by SUN1 and 2 proteins. The nuclear lamina lines the interior aspect of the inner nuclear membrane. Lamin protein filaments polymerize to form a mesh that largely determines the stiffness of the nucleus, in part by tethering chromatin. Chromatin binding is mediated in part by embedded proteins that harbor a LEM domain. EMP1 is a 5-pass internuclear membrane protein with a C-terminal nucleoplasmic putative BAF binding domain. NEMP1 is a 5-pass internuclear membrane protein with a C-terminal nucleoplasmic putative BAF binding domain that binds double-stranded DNA in a sequence-independent manner and interacts with emerin and other chromatin-binding components of the nuclear lamina. In response to tissue stiffness and force exerted upon the nucleus, lamin A, emerin, and NEMP1 modulate nuclear physical properties and transcription, through these nuclear envelope complexes, forces may directly alter chromatin conformation. To distinguish direct effects from indirect force-induced signaling cascades, it would be helpful to measure force transmission across the LINC complex and the internuclear membrane lamina. Here, we present a pair of knock-in FRET-based sensors that permit the measurement of tensions across the LINC complex component Nesprin-2 giant and internuclear membrane protein NEMP1 in mice. The former was previously generated for use in vitro, whereas the latter is an entirely new sensor. Control transgenic strains were generated from FRET donor-only and headless-tailless constructs, and the expressed sensor proteins localized appropriately. We also introduced software that mitigates challenges imposed by autofluorescence and low signal-to-noise ratio to facilitate the fitting of fluorescence decay curves generated by fluorescence lifetime imaging microscopy, FLIM, of living embryos. The sensors respond to cytoskeletal relaxation and externally applied stretch by exhibiting increased fluorescence lifetime/tension means or ranges. By employing the mouse embryo for proof of principle analysis, we identify spatial and temporal differences in tension across the nuclear envelope that correspond to nuclear roundness and tissue stiffness. Results. Subsection: Generation of tension sensor mice. We wish to generate knock-in mouse strains that harbor tension sensors within the outer and inner aspects of the nuclear envelope. To measure force transmission across the LINC complex, we generated a mouse strain capable of conditionally expressing a previously published tension sensor, NESP-TS. The construct consists of a FRET-based tension sensing module between the actin and sun binding domains of Nesprin-2G. A previously described headless control construct that lacks the actin binding domain of Nesprin-2G which is NESP-HL, was prepared as a separate transgenic construct. Since chromatin is a structural component of the nucleus, and chromatin architecture regulates transcription, we considered internuclear membrane tension sensor candidates that link the membrane to chromatin and chose the internuclear membrane protein. A previously characterized FRET-based tension sensor module was inserted between the final transmembrane domain and the C-terminal putative BAF binding domains of NEMP1, NEMP-TS. Control constructs, FRET donor only, so NEMP-DO, and tailless, NEMP-TL, were also prepared. The tension sensor and control constructs were transiently expressed in vitro. Super-resolution stimulated emission depletion, STED, microscopy revealed appropriate co-localization of NEMP-TS and controls with lamin B1, and that of NESP-TS external to lamin B1. Previous findings suggest that in vivo imaging of FRET-based tension sensors is impaired by low signal-to-noise ratio. For this reason, and to avoid developmental stage and tissue expression disparities, we employed a CAG promoter to ensure robust expression of the 5-sensor or control reporters. A lock-stop-lock sequence interrupted the promoter gene junction to allow for Cree-based conditional expression. Constructs were knocked into the ROSA26 locus by homologous recombination in embryonic stem cells, and chimeric mice were generated by embryonic stem cell morula aggregation. Following germline transmission, mice were bred to NLS Cree or CMV Cree mice to generate ubiquitously active strains, henceforth referred to by their construct name. By confocal fluorescence microscopy, we confirmed that all 5 sensors and control proteins localized to the nuclear envelope in vivo. FLIM signal was readily detectable within different tissues and under experimental perturbations in vivo. Importantly, NIMPTS and NISPTS caused no measurable alterations of nuclear shapes compared to H2B-GFP, and comparisons of nuclei marked with either NIMPTS or NISPTS revealed a small difference in nuclear size that is expected based on their relative positions at the inner nuclear membrane or outer nuclear membrane in E9.5 distal forelimb mesenchyme. Adult mice from all 5 new strains that ubiquitously expressed the transgenes were morphologically normal, fertile, and had normal body weight. Nesprin-2 null mice are known to be viable and fertile. The NISPTS can functionally rescue centrosomal orientation and nuclear movement defects of NIH3T3 fibroblasts depleted of endogenous Nesprin-2 in vitro. To test the functional equivalency of NIMPTS to NIMP1, we bred the sensor and control strains onto a NIMP1 null background. Presence of the NIMPDO, but not NIMPTS, transgene rescued the otherwise markedly diminished litter size of females at E8.5 to E9.5. These findings imply the length or nature of the tension sensor domain rather than the level of expression or disruption of the endogenous amino acid sequence most affects function of the protein. We concluded that NIMPTS is not functionally equivalent to NIMP1, but could be a useful measurement tool. Subsection: an in vivo analysis toolkit. Acquiring force data in live mouse embryos is conceptually ideal, but it reflects a physiologically relevant environment. But analysis by FLIM or FRET is challenging due to low signal-to-noise ratio in vivo. Autofluorescence, light scatter, and low photon counts are obstacles to reliable data analysis. To mitigate these challenges, we developed software to semi-automatically segment regions of interest, such as nuclei, and optimally fit fluorescence decay data acquired in vivo. We called the software FLIMvivo. We chose to analyze sensor data using FLIM because the method is independent of fluorescence intensity, unlike ratiometric FRET analysis. FLIM data were acquired at 2-minute intervals under live conditions. To maximize signal-to-noise, nuclear envelopes were isolated from the resultant images using a segmentation manager we prepared for FLIMvivo. Difference in the segmentation efficiency between the 2 tension sensor strains was not significant. The program compiled data across 6 time points to increase photon counts and to improve fit quality for individual nuclei. There are 2 principal sources of noise in our setting: a quick fluorescence decay that is likely due to autofluorescence can artificially decrease the measured fluorescence lifetime, and background or scattered light can artificially increase the fit value for measured lifetimes unless addressed appropriately. In addition, signal-to-noise ratios between samples and tissues cannot be assumed to be consistent. Managing autofluorescence is complicated by the fact that the fast mode directly following the laser pulse is often not much longer than the timescale of the instrumental response function. There is not an obvious way to fit individual curves to account for these problems using a simple tail fit, because it is not clear where each tail should be cut. Furthermore, any given segment generally does not have enough data to simultaneously fit for autofluorescence and FRET signal time constants, as they are poorly correlated. We overcame these challenges by recognizing that autofluorescence is likely unrelated to our sensors, thereby allowing us to fit for autofluorescence signaling using the entire field of view, and by using a full convolution fit that properly defines the instrument response function. Once these features were accounted for, individually segmented regions of interest could be analyzed. Subsection: testing sensor responses in vivo. We tested the individual sensors in vivo using E9.5 wild-type distal limb bud mesoderm as a model system due to its accessibility and well-studied patterning characteristics in intact embryos under live conditions. Relative to NEMTS, NEMTO exhibited higher mean lifetime values as expected. NEMTL and NESPTL reported low lifetime values relative to NEMTO and previously published donor-only MTFP measurements, with low standard deviation as expected. Appropriately, NEMTS and NESPTS reported a wider range of lifetimes than those of NEMTL and NESPHL, but unexpectedly reported lower means compared to their tailless and headless controls. The relatively narrow and consistent lifetime ranges reported by the tailless and headless controls between tissues suggests that, unlike their full-length counterparts, these controls do not respond to tension, but are trapped in a semi-stretched state. It was recently shown that the position of the sensor module within a similar Nesprin 2G construct used in vitro can unexpectedly decrease fret index, but does not compromise the full-length sensor readout. For future applications, we will preferentially compare full-length tension sensor readings to donor-only control means, whereas the tailless and headless controls will be useful for comparisons of lifetime range. We next sought to alter nuclear characteristics in vivo. To test the effect of actomyosin inhibition upon our transgenic sensors, we treated embryos in roller culture with Rho-associated kinase inhibitor Y27632. Compared to carrier-treated controls, Y-compound treatment reduced donor lifetime across both sensors. Inhibition of actin polymerization by treatment with cytoskeletal D produced similar results across NEMTS. It is recognized that cytoskeletal forces are exerted through the LINC complex, and this finding confirms those forces are transmitted across the internuclear membrane. Pharmacological perturbations of global chromatin architecture by treatment with either doxorubicin or 2-deoxyglucose and sodium azide significantly reduced tension across NEMTS. Those data suggest that condensation and fragmentation of chromatin globally relaxes the tension module. To determine if our sensors are responsive to exogenous tensile stress, we applied precisely controlled micro-aspiration of the E9.5 distal forelimb bud, as we have done previously. Aspiration up to 2,500 pascals, but not treatment with Y27632 elongated cells. When aspiration was maintained during FLIM acquisition, mean lifetime tension across NEMTS was increased. For NESPTS, aspiration widened the distribution of lifetime values but did not change the mean. Possible explanations for this finding are that LINC complex tension is a function of perinuclear orientation with regard to the axis of stress, or that the complex remodels to dissipate stress. The two sensors therefore report related but distinct aspects of force transmission under the nuclear envelope. Subsection: measuring nuclear forces in developing tissues. One aim of generating knock-in versions of nuclear tension sensors is to link spatiotemporal changes in morphogenetic forces to gene expression. As a first step towards that goal, we tested the output of our sensors in different tissues and at different times. The heart and brain are potentially useful proof of principle candidates because they are structurally different tissues that exhibit distinct mechanical properties. Both NEMTS and NESPTS embryos reported higher mean forces within the beating E9.5 myocardium than from the E9.5 anterior forebrain. an observation that is consistent with the contractile behavior of cardiomyocytes. Spatial variations may also exist within structurally more comparable tissues. The limb bud is a compelling model system because distinct domains of patterning genes are expressed along its proximodistal axis, suggesting there may be a link between mechanisms that form the limb bud and pattern its differentiated tissues, such as the cytoskeleton. Evidence for mechanical heterogeneity within early limb bud mesoderm includes our earlier findings using magnetic tweezers that the proximal core is stiffer than the distal periphery. Here, using light sheet microscopy to visualize CAG-driven H2B MIRFP703, a far-red fluorescence nuclear reporter in live transgenic embryos, we found 3D nuclear sphericity is greatest in the proximal mesoderm of the E9.5 limb bud. At the resolution of unsegmented tissue, apparent tensions across NEMTS were also graded in a proximal high to distal low fashion, suggesting nuclear sphericity correlates with internuclear membrane tension in this setting. To more directly examine the relationship between nuclear shape and nuclear sensor output, we evaluated nuclear shapes from the same confocal images we analyzed by FLIM. NEMTS tension correlated with roundness, whereas NESTS tension trended insignificantly with elongation. To distinguish fluorescence lifetime along long and short axes of nuclei, we wrote a software supplement for FLIMvivo to segment nuclear envelopes in 2D. Consistent with previous NESTS findings in vitro, neither NEMTS nor NESTS reported significant axis-dependent tension disparities, even under micro-aspiration, raising the possibility that the sensor proteins share the same mechanical load around the nuclear envelope. A caveat to nuclear shape segmentation in 2D is the possibility of underestimating length-to-width ratios of elongated cells depending on the obliquity of optical sections. These data reveal similarities and differences in the responses of the two sensors and establish their ability to distinguish spatial variation of nuclear envelope tensions within a tissue. To test for temporal changes in nuclear forces across development, we examined limb bud mesoderm at E9.5, E10.5, and E12.5 for NEMTS proximal high, distal low patterns of measured lifetimes forces persisted over time. Across those time points, mean NEMTS tensions diminished, while mean cross-sectional nuclear area enlarged, suggesting that NEMTS tension correlates with smaller nuclei. For NESTS, proximodistal variation was not apparent until until E12.5, when a proximal low to distal high pattern was observed. That pattern is not unexpected given the different response of NESTS to cell shape changes that we identified by microaspiration. The spatial variation of the two sensors suggests there's a gradient of forces exerted across the outer and inner nuclear layers of the nuclear envelope at E12.5. Together, these findings suggest that nuclear mechanotransduction is a dynamic and nuanced process. We must contend with these nuances if we are to decode the role of mechanical forces in gene expression and pattern formation. Final section, discussion. The tension sensor mouse strains we generated are complementary to an expanding suite of tools one can apply to examine mechanical mechanisms that drive cell behaviors and regulate gene expression in vivo. NEMTS and NESTS are sensitive and specific sensors capable of detecting exogenous and endogenous tension across the nuclear envelope in vivo. but are not intended to test the specific characteristics of their host proteins. From one perspective, it is advantageous that the sensors are not functionally equivalent to their intact host proteins because the potential for phenotypes secondary to ubiquitous expression is avoided. By combining data from our reporter strains with analysis of cell and tissue properties and forces, chromatin conformation, and RNA and protein expression at single-cell resolution, it is now possible to test mechanotransduction hypotheses in a quantitative fashion in vivo. FRET controls do not necessarily behave as expected in vivo. In particular, headless-tailless sensor constructs do not uniformly report maximal FRET, minimal FRET values, and would reflect a fully relaxed spring within the tension sensing module. Possibilities to explain those findings, such as module position-dependent effects, have been tested previously, and may also include unanticipated attachment of the headless-tailless protein and to other proteins or nearby membranes through lipophilic regions. By identifying the spatially independent output of tailless-headless controls, we were able to distinguish the spatially dependent changes in the range of the full-length sensor constructs. The significantly lower variation of lifetimes generated by the tailless and headless controls validates the actuation of the sensors under endogenous forces. Although the availability of transgenic control strains was very useful to define the specificity of the sensors, their spatially stable behavior implies that comparison to controls is not necessary for all future analyses, especially within a given tissue type. While testing their responses to perturbations, we observed overlapping and distinct outputs between the two sensors. As expected, measurements using the Nesprin 2G and NEMP1 tension sensors confirmed that cytoskeletal forces are transmitted through the LINC complex and the nuclear lamina. Those observations are in keeping with our finding that NEMPTS is under greater tension in relatively spherical nuclei, since that trait corresponds to higher cortical tension. Interestingly, forced nuclear stretching resulted in distinct responses by the two sensors. Unlike the NEMPTS that responded in a relatively linear manner, the NESPTS exhibited a greater range of values. One possibility is that the stress-induced cytoskeletal remodeling results in a relatively broad range of NESPTS values. The overall strategy we employed has pros and cons. Use of FRET-based sensors is advantageous since the output is largely not dependent on the expression levels. Major differences in expression are avoided by the strategy of knock-in to a ubiquitously expressed single genomic site. However, the in vivo nature of our analyses presents particular challenges. Poor curve fitting, resulting from the combination of low photon counts and background fluorescence, autofluorescence, and background scatter, was a barrier to reliable FLIM analysis when we applied commercially available fitting software. Similar hurdles were encountered previously with an E-cadherin tension sensor in Drosophila, resulting in apparently poor sensitivity. Despite our tissues of interest being deep within the mouse embryos, the software we developed through multiple rounds of iteration permits reliable and consistent FLIM analysis in living tissues and is applicable to a broad range of FLIM analyses. The differences we observed between tissues such as the brain and the heart, as well as along the proximodistal forelimb bud axis, correspond to previous measurements of tissue stiffness. Those correlations imply that traction and contractile forces exerted by cells in stiffer environments are transmitted to the nuclear interior. Since gene expression domains co-vary with these properties along the proximodistal limb bud axis, we intend to examine the influence of forces upon transcription. Mechanical effects of force transmission to the nuclear interior is an exciting frontier in developmental and cell biology. Our ability to measure force transmission in animal systems opens doors to explore this area, and future technologies will no doubt improve the variety and precision of tools that measure how forces are transmitted to the nucleus.